Okay, so let's discuss what is biotechnology. The definition for biotechnology in our Campbell Bio 1010 book is the manipulation of organisms or their components to produce useful products. And so this picture here is actually showing you some of the things that have to happen in order for biotechnology to work. So we need things like host cells, we need cell culture, we need to be able to extract DNA, we need to be able to manipulate DNA in foreign places and then insert it into cells so those cells can actually grow up and produce the gene of interest that we want. So we need to deal with tissue culture or cell culture, and then we also need to analyze our product to make sure we're producing the product that we want. So I'm going to go over some of the techniques of the last 20, 30 years that allowed us to get to this point. So the first one is, remember that Watson and Crick discovered the structure of DNA. Well, they discovered it in the sense that they proposed this model for the structure of DNA based on the work of Rosalind Franklin and her X-ray crystallography. And in 1953, they published a short paper and were awarded the Nobel Prize. Sadly, Rosalind Franklin had passed away, probably due to cancer that she got from working with all the X-ray crystallography that she did. So she basically, her contribution to this field also resulted in her death, early death, and she was not part of the winning team. So we also have to remember that the way cells work. And in 1961, Francois Jacob and Jacques Monod were the ones who helped come up with this idea of a genetic code. And as we already discussed in class last time, we talked about how DNA gets made into messenger RNA. That messenger RNA is modified, then it leaves the nucleus through a nuclear pore, and in the cytoplasm it is actually translated into a protein. So we have to know about this in order for it to work. What the amazing thing is, the work of Gobind Khorana and Robert Holly, who were the people who came up with this genetic code. And they got a Nobel Prize for their work where they figured out the combinations of the four DNA bases, how they turn into amino acids. And we did a little bit of work of this in class. So this genetic code is really the Rosetta Stone of life. And this genetic code is universal, whether you are a bacteria, a virus, or even as complicated as a human, which we like to think we are, we all use that same genetic code. If there is another genetic code on Earth, it doesn't work anymore, or we have not discovered any living organism yet that does not use this code. We talked about that in class. So probably one of the biggest discoveries that got recombinant DNA going was the discovery in the 1970s of restriction enzymes. And restriction enzymes are enzymes that are found in bacteria, and it's basically an immune system of the bacteria. Bacteria have this problem in that they constantly get infected by viruses. And so by having a restriction enzyme, when a virus comes along and puts some of its DNA into that bacteria, the bacteria's got an enzyme that can go and chop it up into smaller pieces. And so different bacteria have different restriction enzymes that cut specific sequences of DNA. So here it is an example of a restriction site. And what happens when the enzyme runs along the DNA and finds this distinct pattern of G's and A's and T's and C's, it cuts between the sugar phosphate backbone and generates what we call a sticky end. And so just by seeing this at the DNA level, it kind of makes sense that you could actually take these sticky ends and mix and match them with other DNA molecules and make recombinant DNA. So here you can see two types of DNA both cut with the same restriction enzyme, and then they mix together, and every now and again you're going to get what we call a recombinant, when a piece of one organism's DNA is inserted into the other organism. And then of course we need another enzyme called DNA ligase, which is kind of like the glue, which now will join that sugar phosphate backbone and seal up the gaps. And now you have a recombinant DNA molecule.
Restriction endonucleases are enzymes that cleave DNA at specific nucleotide sequences. The sequence recognized is often 4 to 6 nucleotides long. For example, the restriction endonuclease EcoR1 recognizes the sequence GAATTC. The nucleotides at one end of the recognition sequence are often complementary to those at the other end. The two strands of the DNA duplex have the same nucleotide sequence running in opposite directions for the length of the recognition sequence. Because the same recognition sequence occurs in both strands of the DNA duplex, the restriction endonuclease can bind to and cleave both strands of the DNA molecule. Because the bond cleaved is typically not positioned in the center of the recognition sequence, and the DNA strands are antiparallel, the cut sites are offset from each other. After cleavage, each DNA fragment has a single-stranded end a few nucleotides long, and the single-stranded ends of the two fragments are complementary to each other. These single-stranded ends can pair with each other, sticky ends. Once their ends have paired, the two fragments can be joined together with the enzyme DNA ligase, which reforms the phosphodiester bonds of DNA. What makes restriction endonucleases so valuable is that any two fragments of DNA produced by the same restriction endonuclease can be joined together. Restriction endonucleases are fundamental tools in genetic engineering.
So that animation is a great animation just showing one restriction endonuclease. In fact, there are countless restriction endonucleases out there. They just are isolated from different bacteria. The very first one came from our friend E. coli, but not the strain we use in the lab. RY13 was the strain, and it was the first one identified, so it was known as EcoR1. There are many other restriction enzymes out there, each with unique recognition sequences.